This is chapter 11, which deals with microbial genetics. Remember that DNA contains all of the genetic information for an organism, including regulating protein synthesis. Because remember, proteins are made up of a chain of amino acids. How do you know which amino acids to put together for which protein? That information's on the DNA, so everything goes back to the DNA. The central dogma just states the way things flow, the information flows. So it's going to start with the DNA, move to the RNA, and then finally to the protein. One thing you may hear about in any biology course is the unity and diversity of life. And what this is referring to is the fact that a lot of the building blocks, if you will, are the same. So when we're looking at, say, DNA, and this is going to hold true for RNA and proteins as well, the building blocks are the same, whether you were talking about a bacteria, a fungus, a human, a plant. It doesn't matter what the organism is. They all have the same principles. They all have the same building blocks. So for DNA, they all have the phosphate group. They all have the sugar deoxyribose. They all have those same 4 nitrogenous bases, A, T, C, and G. It's how you put those together. What order do you put those nitrogenous bases in for the DNA sequence that makes the difference? For amino acids, there's 20 amino acids. How do you put those 20 amino acids in what order to make a protein? The 20 amino acids are the same for any protein. And regardless of the organism you're talking about, it's how you put those together that you get the differences. An analogy, and I've done this before, where in a class, obviously a face-to-face class, I give all the students a box. So if I were to give everyone a box and you look in it and everybody has the same thing inside that box, it contains 100 Legos total. You've got 25 blue, 25 green, 25 red, and 25 yellow. So everybody has the same starting building blocks. And I just say, build something. What do you think is going to happen? Everyone has the same building blocks, but how you put it together is going to be different. Some people are going to make a real tall column. Some people would put, say, all the blue ones first, then all the green ones, then all the red ones, and then all the yellow ones. Some people may do a column as well, but instead they're just going to put one blue, then one green, then one red, then one yellow, and then alternate them. Or maybe the order of the colors will be different. Some people might make squares. Some people might make a pyramid. Some might make a rectangle. The point is, there's going to be a variety of things built. But you all started with the same building blocks. So the unity is that every organism starts with the same building blocks for DNA, for RNA, for proteins. But it's how they are put together. That's where we see the diversity. That's where we see the differences. Once again, the central dogma refers to the flow of the information, and that you start with DNA. DNA contains all that central information. Through the process of transcription, DNA is used as a template to make RNA. Through the process of translation, RNA is used as a template to make proteins. And sometimes people will confuse transcription and translation. And so what I usually say is this: DNA, let me put this a little bit darker here. So your DNA, and I can no longer see where I'm at. Here we go, DNA. And it will help if I change the color. That's what I'm trying to do, is mark this so you can see it. There we go. DNA, the building blocks are those 4 bases, the nucleotides. It's a nucleic acid. RNA is also a nucleic acid, which means their building blocks are the same. In other words, they speak the same language. What do you do when you transcribe something? What you're doing is you're simply copying from one point to another. You take the information that's on DNA and you copy it directly over to RNA. You're using the same language. They're both nucleic acids. Translation. When you translate something, you're translating from one language to another. Well, you're starting with RNA, which is a nucleic acid, has those nucleotides, the 4 bases, etc. Proteins, the building blocks now, are amino acids. That's a different language. So you're going to have to translate, take the information from RNA and translate it to make the protein. Translation, you're changing languages. It's like on the DNA, you have the information. Hi, how are you? You just transcribe. You take that same information, you put it on the, copy it over here to the RNA, and you make that RNA. Hi, how are you? That's transcription. Translation, now you're taking that hi, how are you on the RNA, but to make the protein, you have to switch to a different language. And now you have to go, oh, bonjour, comment allez-vous? You change languages on there. And there's a way that you're going to see that allows for this. It's kind of like having a translation dictionary with you. With genetics, remember genotype, that refers to the genes that are present on the DNA. Phenotype refers to what you physically see. What you physically see, that phenotype, that is going to be determined by the genotype. And it may be influenced somewhat by environmental conditions. Are you starting to get an idea how everything relates back to what's on the DNA? So sometimes, and we see this with a certain bacteria, I believe we mentioned before, where there is a gene for a pigmentation of a particular bacteria, Serratia, depending on the environmental conditions. In this particular case, example, it would depend on the temperature. If it's at room temperature, you get one color. If it's at a higher temperature, you get a different color. So the gene is ultimately on the DNA in the organism, but environmental conditions can play a role as to what you're going to see. So with the central dogma of starting with the DNA, making RNA, and then finally making your protein, we're going to go and discuss those processes now. So we're going to start with DNA replication. DNA replication, some people refer to it as DNA synthesis or DNA duplication. You're making a copy of the DNA. You're going to have to make a copy of the DNA before the cell can go through cell division, because when it goes through cell division, each new cell has to have its own copy of the DNA, which means you have to make another copy of the DNA before cell division. So this is what we're going to do. DNA replication is referred to as semi-conservative. What that means is DNA is double-stranded. Those two strands are going to have to separate, so each strand is used as a template to make new strands. Both of them, you get 2 new strands made. Semi-conservative means that when you are done with the replication process, the 2 original strands do not go back together, and the 2 new strands do not go together. Semi-conservative means that when you're completely done, you have 1 original strand and 1 new strand. And then what happened to the other original strand? It pairs with the other new strand. It's going to require a lot of different enzymes. It's going to be highly regulated and controlled. It's going to start at a certain point. It's known as the origin of replication. It's kind of like, here's a big signal. That's where we start. Prokaryotic cells only have 1 origin of replication site. Eukaryotes will have multiple sites. And it's also what we call bidirectional. That means at this origin, you can go right and you can go left. And that's what will happen, is that you will start with DNA replication, but it's going in both directionals. So that's why we say it's bidirectional. So you're going to make use those parental strands as a template and making the new strands, which we refer to as daughter strands. How are you going to make it? You're going to have that backbone being made, and you're going to add 1 nitrogenous base at a time, and you're going to use what's known as the complementary base pairing rule. That's how you know what base to add for the new strand. And what does that mean? Remember, A always binds with T, C always binds with G. So wherever you see an A, you're going to put a T. Where you see a T, you're going to put an A. If you see a G, you put a C. If you see a C, you put a G. So this is going, like I said, it's going to involve a lot of enzymes. Just kind of work through this. We start with initiation. There's a message that comes, oh, okay, we got to replicate the DNA. One of the first enzymes involved is known as DNA gyrase. It's also known as topoisomerase 2. And its whole purpose, remember, DNA is in a helical shape, so it's twisted. You have to unwind it before you can separate the strands. And so that's the purpose of the DNA gyrase, is to unwind that DNA. Helicase is going to be the next enzyme that comes in now that you've unwound it, and it's going to separate those two DNA strands from each other. And where they're separated at that point is referred to as the replication fork. Now, single-stranded DNA is a little unstable, and it's a little susceptible to being broken down. So you're going to have some proteins that are going to bind to the single-stranded to help stabilize that DNA. Then you're going to have RNA primer that comes in. There's a primase that makes an RNA primer that's going to start the process. The main enzyme is going to be DNA polymerase 3. There are a couple of different DNA polymerases. DNA polymerase 3 will begin, but it can't initiate the process. You have to have this RNA primer just to get started, then the DNA polymerase can begin. And DNA polymerase can only work in one direction, the 5' to 3' direction. This is referring back to when we discussed the structure of DNA and the sugar, the deoxyribose. If you remember, it has 5 carbons, and we number those carbons in chemistry. That's what the 5' and 3' are referring to, are the carbons on that sugar molecule. And we didn't really get into it, but it just has to do with the orientation of the sugar. So you can look at a strand and you can figure out, okay, is this the 5 or is this the 3? And the bottom line is, remember the DNA strands, we've got the 2 of them, that we said they're anti-parallel, so they're going in different directions. This is going to play a role now because DNA polymerase 3 can only work in one direction. So how's it going to handle this? So I know this is a confusing slide. Just deal with this. For those of you that are not familiar maybe with PowerPoint, if you're watching this on PowerPoint, when you are in the slide mode, you can enlarge. If you right click, you can enlarge it. Also, all these pictures are from your book. So I know on here is trying to find a picture to have everything in it, but I know it looks a little confusing. So here we go. Here's DNA replication. So on this end, you have your double-stranded DNA, that light-colored, pale blue color. Those are your DNA strands. So this is your point of origin where the replication fork is right here. You've got the helicase that is separating the strands. You have those little circles are your proteins that are involved with stabilizing, and then you've got your RNA primer. Now, you are making the DNA in this direction. I'm sorry, I know I'm not real steady with drawing. That's why I'm not an artist. So what happens is you've separated the strands. You would have started. This is a continuous strand. The red is a new DNA strand that's being made. It's using the existing strand as a template. And so you're adding on, way up here, you're adding on one base at a time, and it's just going to keep moving. This keeps opening up, and then at the replication fork, and the red arrow is going just to continuously add one new base at a time. This is referred to up here on the top is the leading strand. It's made continuously. It's just making one base at a time. Like I said, if it sees an A, it's going to put, if there's an A right here, it'll put a T right here. If there's a C right here, it's going to put a G right here. So one base at a time, it's adding. It's just zipping along. Now over here, because you've got your numbers here listed, it can only read 5' to 3', so it has to read this direction. This strand is going in the opposite direction because they're anti-parallel, which means, as you can see from the arrow right here, this is the direction that it's being made. So what happens, but it's opening in the other direction. So you have to do things a little bit differently. You're going to use this as a template, and you're going to make a section. Now you open up some more. Oh, now I can make this little fragment. Oh, this is now open. Now I can make this fragment. Use this as a template. So you're making these little fragments here. Once this finishes, now you're opening up over here, so it's going to be able to come back over here and make a segment here. It's making a segment here. You're making all these fragments. These fragments are known as the Okazaki fragments, named after the person that discovered them. You can't have DNA as fragments. So the original strand is solid that you were using as a template. So you're going to have this other enzyme called DNA ligase that's going to come and connect the two pieces together. So you're showing here, right here, connect. I often kind of jokingly refer to DNA ligase as kind of like your DNA superglue. It's going to connect these fragments all together. So eventually at the end, you will have, up here, you will have the original strand, new strand. Down here, you're going to have the new strand and the original strand. So you find the origin of replication site. That's where you're going to start. You have your gyrase come in, untwist the DNA helicase, separates the two strands. You put the stabilizing proteins on, and then the RNA primer starts the process. Then DNA polymerase III is going to come in. It's going to read the original DNA strands, use that as a template, and it's going to be adding in one base at a time, making the new strands. You're going to have a continuous strand. You're going to have what we call a lagging strand. That's the one where it's made in fragments, those Okazaki fragments, that then have to be connected by DNA ligase to connect all those fragments together. There have been studies that have been done to try to measure how fast this is going. DNA polymerase III is adding, I've seen anywhere from 1 to 2,000 bases per second. It's moving. It also acts as a proofreader. So as it's adding a base in, if it say is putting, oh, here's an A, put a T. Here's a C, put a G. Here's a G, put a A. Oh, wait a minute, put the wrong base there. And it'll back up and it'll fix the problem. Does it catch all the problems? No, it does not. But it does a pretty good job. As you can see in this diagram up here at the top, you had your point of origin here, and when I say it's bidirectional, as you can see, you're moving in two different directions, and you end up with, this is often referred to as a replication bubble, because it looks like a little bubble there, but you're moving in both directions. That is DNA replication. So, as I said, the initiation is getting all the players in there, unwinding the DNA, untwisting it, separating the strands, stabilizing it, get the RNA primer in there, and then the DNA primer. Elongation is where you're adding those bases one at a time. Like I said, it's anywhere between 1,000 to 2,000 bases per second, depending on which report you read. You have a continuous strand, which is the leading strand. The non-continuous strand, that's the lagging strand, because it's being made in fragments, those Okazaki fragments. The DNA ligase binds the Okazaki fragments together. Then you're going to have DNA polymerase 1. This is a separate enzyme that comes in and it removes the RNA primer and it's going to replace it with DNA. And then termination in prokaryotic cells, it's when you end up with the two circular DNA pieces separated from each other. Eukaryotic DNA is linear, and so it just goes to the end and it's done. Telomeres protect the genes near the ends of linear DNA. There's going to be a repetitive non-coding section, and so each time it gets replicated, a little piece of that non-coding section is not going to be coded. Basically, it gets progressively a little bit shorter every time it replicates. So this is showing with eukaryotes, you number one have multiple sites of origin, and so you start in one place, and this is just going to continue until they meet each other. RNA transcription. This is where you've gotten a message that you need to make a protein. So how are you going to do that? Well, to make a protein, you need to have a messenger RNA that contains the information of which, like amino acids you're going to need. Where does a messenger RNA come from? Well, it has to get that information from the DNA. So are you going to read the entire DNA? No, you just need a small segment. If you just are making a protein, you just need to get that information from that one gene. Remember, DNA has thousands of genes on it, so you just need a little tiny section of it. So there's going to be an area of DNA that you're going to have to separate the two strands so you can read it. When you separate them, it's going to form this bubble. The DNA strand is going to be used to make, as a template, to make the messenger RNA or the mRNA. In this case, you're going to use the enzyme RNA polymerase because you're making RNA. Similar to what we saw with DNA replication, RNA polymerase is going to come in, it's going to add one base at a time to make that mRNA. Now, transcription is going to be very closely regulated because if you don't need the protein, why make it? So RNA transcription is divided into 3 steps. You have initiation, elongation, and termination. In initiation, RNA polymerase is going to bind to an area on the DNA that's referred to as the promoter site. This helps to orient it, to position it. Do you know which way you're going to read? Do you know where you're going to read? Elongation is when now you are adding one base at a time. Once again, you're using that complementary base pairing rule. You have to remember, though, C and G go together. If it sees a T on the DNA strand, it's going to put an A. But remember with RNA doesn't have thymine, doesn't have T. Instead, it has uracil. So wherever it sees an A on the DNA strand, it's going to put a U for uracil. Just keep that in mind. So it's going to be adding one base at a time. It's a little bit slower, a lot slower. Excuse me, a lot slower than DNA replication. It's adding about 40 bases per second. The DNA it's constantly unwinding and rewinding. So it's going to unwind and you make a bubble where you're reading. And just think of that bubble as moving along. It's kind of like you're unzipping and the zipper got messed up, so there's an open there. So this bubble's going to move along. We'll see it in the picture in a second. Termination is the final step. This is when RNA polymerase reaches the end where there's what we call a termination signal that basically is saying stop, and all the components release and the DNA rewinds back up. So in this picture for transcription, the light colored bluish is the DNA. So you would have had your promoter site, it would have started you in well, it's showing right down here. This is where you would have started everything. So you would have bound, the RNA polymerase would have bound here, and that would orient it to say, you need to go this way. Not the other way. You need to go this way. Come on, let's start this way. And so, as you can see, it's what it's doing. So it started right here. This is the bubble area that we talk about, where it's unwound. As it moves along, once it has round a section, this part of the DNA, it rewinds. It's opening and unwinding over here. Excuse me. Now, on here, what is happening is the, and there we go. Sorry about that. So it's unwinding in this direction. So you're moving this way. You are adding one base at a time as it's moving along here. So right here, there's a C, it put a G. Here's an A, it put a U. Here's an A, it put a U. Here's a G, it put a C. Here's a T, it put an A, etc. That base pairing rule, that is the way that it's going. This red strand is your mRNA strand. And so the end of it here gets detached. When it reaches the termination point, then this entire strand will be released, the red strand, the messenger RNA. The RNA polymerase will leave. The bubble will go back together, and so it's fine until the next time you need to make a protein. So there's initiation, elongation, and termination. In eukaryotes, there's a couple of additional steps. So after termination, and like I said, this is only in eukaryotes, you're going to add a cap at the 5' end. That just means you're adding a G, a guanine. This helps for protection, and it's going to actually help initiate translation in the next step. You're also going to add what's known as a tail. On the 3' end, you're going to add what's known as poly A. It's about 200 adenines. This is also for protection. Then you're going to go through a process called RNA slicing. This is where you're going to remove parts of the mRNA, some of the bases, known as introns. And then the remaining ones are known as exons. You splice those together, connect them together using your ligase again. Eukaryotic DNA has introns in it. These are sections of DNA. What it is, you have a long strand of DNA. And so when you're going through transcription, you have the promoter site, say right here, and here's the termination site. So you, during transcription, you're going to copy this entire piece. However, you don't need all of the pieces. Maybe for this particular protein, if I divide this up into sections, so 1, 2, 3, this is supposed to be a 3, guys, 4, and 5. So, maybe for the particular protein that you're wanting, you only need section, we're going to change colors again here. Say you only need 1, 3, and 5. So 2 and 4 would be your introns. Those are the pieces you don't need. So when we're talking about the slicing, you're going to cut out, in this particular for the green, you would cut out 2, you would cut out the 4. You're doing it on the strands here. You cut this out, and then splice the remaining pieces together. Why do eukaryotes have this? Because in this one section, you've got these 5 different pieces. Well, for the protein right now, you need 1, 3, and 5. But maybe down the road sometime, you might need a different protein that's going to use 1, 2, and 4. And then there might be another protein sometime that you're going to need that would use 2, 3, and 5. You start to see how mathematically you've got 5 fragments. How many different combinations can you come up with? Instead of having a much longer DNA in this one section, you've got these 5 different fragments, but you can come up with a huge number of variable combinations, and all of them would be for different proteins. So that's the advantage of having them. Prokaryotic cells don't have this, so they don't cut anything out. But the eukaryotes do. So the introns are removed. You splice together the remaining exons. Now the mRNA can move from the nucleus out into the cytoplasm. Because in eukaryotes, remember, if you're using DNA as a template to make something, the DNA is in the nucleus. So transcription is occurring in the nucleus. Prokaryotic cells do not have a nucleus, so everything occurs in the cytoplasm. So now you have your mRNA made. Now we're going to move on to translation, which is protein synthesis. You're going to use that mRNA strand that you just made. You're going to use that as a template to make the protein. It contains all the information. This is where you have to have your little translation dictionary, because you're going from nucleotide on the nucleic acid on the RNA to amino acids. The way the translation works is it involves what we call codons. A codon is every 3 bases. Every 3 of those nitrogenous bases is equal to 1 amino acid. This is once again universal. It's called the genetic code. It is universal. I don't care what organism you are talking about. It is the same. There are 64 codons. 61 of them code for amino acids. There's only 20 amino acids, so there's a lot of duplication here, as you'll see in a second. 3 of the codons do not code for any amino acid, and we call those the stop codons. Now, some books will talk about the wobble position, and all that's referring to is that 3rd position of the codon, that 3rd base position, is not quite as critical as the first 2. And this is also universal in the AUG. That is the start codon. It codes for the amino acid methionine. In all organisms, that is always going to be the start codon. So here's a table showing the 64 codons. Do you have to know them? Absolutely not. This is not a genetics class. So when I talk about the wobble effect, what that's referring to is, if we look right here, let me get a color, I guess, so you can see better. See how this works. There we go. Right here. The 3rd position. And for this particular case, LEU stands for the amino acid leucine. The 3rd amino acid, if you miss, the 3rd nitrogenous base on this amino acid, if you screwed up and you were supposed to have a U and you stick an A in there in that 3rd position, it's not going to matter. You're still going to end up with the amino acid leucine because there is so much replication in here. That's what the wobble effect refers to. Now, is this always true? No, not always. Over here you can see for histidine and lysine, yeah, it's going to make a difference. If you were supposed to put a U and you put a G, yeah, here you're going to end up with a different amino acid. But you can see here, every 3 bases is equal to 1 amino acid. In translation, you're going to have your ribosomes. Ribosomes are the site of where protein synthesis occurs. So translation is protein synthesis. Ribosomes are composed of two subunits. Now, normally they're separated. They will only come together during this process of translation. Remember, your prokaryotic and your eukaryotic ribosomes are different sizes. Just kind of keep that in mind. In prokaryotes, translation can begin before transcription is finished because they're occurring in the same location. There is no nucleus. A polyribosome or a polysome is when you have one mRNA strand, but you have several different ribosomes attached to it with various stages of the translation occurring. That's much more efficient that way. So over here, excuse me, you can have your DNA, you've got your transcription occurring. So here, this purple reddish line here is your mRNA. You're making the strand. Before you've even finished making it, the end that's already finished, this is where you've started translation. These little things here, those are the ribosomes attached. You already have several, you've got 3 of them attached here right now. With various stages of the protein synthesis occurring. So once again, that's possible in prokaryotes, but not in eukaryotes. Why would you have several ribosomes attached to one mRNA? Because you need a lot of the protein right now. Think of it this way. What would make more sense? If we had a class of, say, 20 students, and I said, okay, we're going to do a community project, and we are going to make little gift bags. We're going to fill them with candy. So I'm going to give each one of you in this class of 20, I'm going to give each one of you a different kind of candy. And I'm going to pass out a bag. And we're going to pass that bag around the room. As it gets to you, you put your type of candy in it. And then, you know, we have to make 500 of these bags. We've got to fill them. So we're going to do this. Would it make sense for me to pass a bag to the first person? They put their piece of candy in it. For me to wait to hand them the second bag, to wait until all 20 people put their piece of candy in and then pass out the second bag? Or would it be much more efficient is once the first person has put their candy in the bag, they turn around, pass the bag to person number 2, then I give person number 1 the second bag. We're going to move a whole lot faster that way. And so it's the same thing with the cell. Don't wait until one ribosome is completely finished making a protein before you start the next one. They're all in different stages of it, but you're going to make the protein a lot faster this way, and you need it right now. So with translation, this is also going to involve your transfer or tRNA. This is a single-stranded RNA, but it's folded. It's often described as kind of being a cloverleaf shape. One end of it is what is called the anticodon region. This is where the 3 bases, if they're complementary, they will bind to the codon region of the mRNA. It's where it's going to attach and bind to the mRNA. The opposite end of the anticodon region is known as the amino acid binding site. It's very specific. It can only bind one type of amino acid. If it binds the amino acid methionine, that's the only one it can bind. It will ignore the other 19. So it's very specific. And so this is showing in red, or pinkish down here, once again, your mRNA. This bluish color here is your tRNA. Right down here is the anticodon region, where it is binding to the codon. At the opposite end right here is the amino acid binding site. That's where the amino acid is going to attach. There's different ways of drawing this. This one under A on the far left is probably the most traditional classic drawing of it. So, initiation. You're going to have 3 steps with translation. There's going to be initiation, elongation, and termination, just like what we had with transcription. So in initiation, what happens here is the anti-codon region on the tRNA is going to bind to the codon region of the mRNA. So you've got anti-codon binding to codon. The small subunit is also going to come in and bind to the mRNA. Then the large subunit will come in, because remember they're usually separated. The large subunit comes in and it's going to place your tRNA in an area known as the P site. So you're putting all the pieces together. So you're going to start here with initiation. Once again, this color coding is thankfully the same, and that the pink over here, you've got your mRNA. You're going to have your small subunit, your large subunit, and then your tRNA. The tRNA is going to come in. How does it know where to bind? Look at the basis right here compared to right here at the start. They are complementary. A and U are going to bind together for the first spot, second spot, once again, A and U, and then C and G. If it did not match up completely, it would just bounce off. That's why the other tRNAs aren't going to bind to it. The little circle here on the end is the amino acid methionine, in this case, that it's attached to. So your tRNA is going to come in, bind the anticodon to the codon region. The small subunit's going to come in, as you can see down here now. The tRNA has bound. Here's your tRNA bound to the messenger RNA, anticodon to codon. Your small subunit came in, and then the large subunit came in and bound. Now your ribosome is together. The two small units are now attached. That is initiation. Elongation is now where the empty site, remember the first tRNA went into what's known as a P site. The site right next to it is the A site. So that's where your second tRNA is going to come in and bind. How does it bind? The anticodon binds with the codon. If they are not complementary bases, they're not going to bind. This brings those two amino acids that are on the opposite ends of the codon. Those two amino acids are now close enough they can form a peptide bond. So the whole idea is you're trying to bring in amino acids. Remember, you're making a protein, which is a chain of amino acids. How do you connect the amino acids together? Well, you got to get them close enough together to form this peptide bond. How do you know what order to put them in? That information is on the mRNA. So you brought in the second amino acid, tRNA with the amino acids. They're close enough together. They form a bond. Whenever you form a bond, that's going to cause a conformational change, shape change. When that change happens, when that bond between the two amino acids form and you get a shape change, that is going to break the bond between the first tRNA and its amino acid. And so that first tRNA, it's going to move from the P site to the E site and then be released. What does it do? Go pick up another amino acid. The ribosome shifts by one codon. So the second tRNA that was in that A site, it shifts to the P site. Now the A site's empty for another tRNA to come in with its amino acid and binds. When now you have the second and the third tRNA right next to each other, those two amino acids are going to be close enough to form a peptide bond. That's going to trigger release of the second tRNA from its amino acid. The second tRNA now goes to the E site and then gets released from the ribosome. Go pick up another amino acid. The third tRNA is sitting in the A site, but the P site's now empty, so it shifts to the P site. The ribosome shifts by one. Now the A site's empty. Another tRNA comes into the A site. Amino acids form a peptide bond. P site tRNA gets basically kicked out. A shifts to P. A gets filled. Amino acids form a bond. P gets kicked out. P is empty. A shifts to P. A is empty, and comes another tRNA. Form a peptide bond between the amino acids. P gets kicked out. A shifts to P. A is empty. A gets filled. Form a peptide bond between the amino acids. P gets kicked out. A shifts to P. This continues on. This is why it's the amino acids are being added one at a time. So this is excuse me, showing here where you form the peptide bond right here between these two amino acids, and then there's going to be a shape change, and so this one shifts to here and then will be kicked out. This one shifts to here, is empty, and you just keep adding one at a time. You have now your growing chain of amino acids, which is chain of amino acids is a protein. That's protein synthesis. This continues until you reach one of the stop codons for which there is no amino acid, and that's when everything is going to stop. There's no amino acid for it, and so what happens for that codon? There's no tRNA to come in, and so what will happen is the two ribosomal subunits will separate from the mRNA. Basically, all the components separate from each other, and you're done. So that protein is released. It can now be used. Now, that protein, there may be some modifications. Maybe it has to add a sugar to it or a lipid to it. That usually occurs in the cytoplasm. So, differences between prokaryotic cells and eukaryotic cells. Because prokaryotic cells do not have a nucleus, that is why transcription and translation can occur at the same time. Eukaryotes, because they do have a nucleus, transcription occurs in the nucleus, and then the resulting mature mRNA, after you've done all the splicing, etc., moves out of the nucleus into the cytoplasm, and translation has to occur in the cytoplasm for eukaryotes. So you notice there's some differences there. Mutations. Mutation is just a change in the DNA sequence. There's different types of mutations. You can have what's known as a point mutation, where you change just a single base. A silent mutation just means you keep the same amino acid. Remember that third position where I said sometimes it's not going to make a difference? That would be a silent mutation. Missense mutation, you end up with a different amino acid in just that one spot. Nonsense mutation, the change created a stop codon. That's going to really mess you up because you're not going to end up with your protein then. Frame shift mutation, this is where you either add an additional base or you delete a base. Insertion, you add an additional base. Because with codons, it's every 3 bases is 1 amino acid. If you add an additional base from that point on, it's going to alter the codon sequence, which means it's going to change the amino acids. Deletion, if you delete a base, that's also going to alter the codon sequence, and that's also going to change the amino acids from that point on. So this table just shows some of the examples of frame shift and point mutations, how it can affect the change or not affect the change in the case of a silent one. Frame shift ones, that's going to be devastating because you change the whole sequence from that point on, and there's no way you can end up with the protein you're supposed to end up with. What are some of the causes of mutations? Sometimes they're just random. There's some things that can definitely increase mutations. Certain chemicals can cause mutations. Radiation, ionizing radiation like X-rays, non-ionizing radiation like UV light. We actually do an experiment in our microlab where we expose a particular bacteria to UV light to try to cause mutations. As I mentioned when we were talking about DNA replication, DNA polymerase does have that proofreading ability. It can repair some damage, but it does not catch everything. So there are different ways of in the laboratory testing to see or identify if there is a mutation that has occurred. Auxotrophs are bacteria that have a mutation that require a specific nutrient, and this is often done purposely in the lab that we can tell, basically distinguish the bacteria that's mutated versus the original wild type, is you make it a specific nutrient requirement for it that you have to add to the media in order for it to grow. Replica plating is a process that you can use to identify the auxotrophs. You basically take your sample specimens and you plate it on 2 different plates. One that has a complete medium, so it has everything that's needed for the mutant to grow. And then the second plate has whatever that specific nutrient, oftentimes it's an amino acid, but whatever that nutrient is, you have it missing in the second plate. And so the auxotroph will not grow on that plate. And it's very easy to compare that to the complete one. Oh, in this position, there's the bacteria, but on the second plate, there's not. That's the mutant. The Ames test. This is a standard test that's used for testing auxotrophic bacteria. We use it to identify potential carcinogenic chemicals. Carcinogenic meaning cancer-causing. So this shows the using the replica plating. It's kind of fun. I've done this before. You take this cover, which is sterile. When I've done it, it's pretty much kind of like velvet, or I've seen where they use Velcro. And here is your original plate. And so this has everything growing. You're wanting to see if there's a mutant in there. So you just take this and you just carefully touch it on the surface. Then you're going to take it. Now, the trick here is you have to remember to somehow mark the orientation. So you're going to mark somewhere on the plate. You can see they put a mark right here and a mark right here. Because however you touch down here, you have to keep this in the same orientation. And so in this case, what they have is, here's your complete medium on the top. You touch this on this plate. You touch it on the second plate. Right here, as you can see, it says this is lacking histidine. And now you incubate these plates, and now you look at them. So you go, okay, if you look right here, there's a colony growing, but it's missing right here where they've circled. So this is where it did not have the histidine. That's the chemical that it needs to grow. It was up here on the top, but not on the bottom. And so there's your mutant right there. So you say, okay, that one right there is the mutant. You can always compare back here to the original plate, and it's this one right above the marker. The trick for this, like I said, I've done this in a class before. It was kind of, I don't know, I thought it was kind of fun to do. But the trick was making sure that everybody kept the correct orientation of the Velcro so you can go back and figure out which colony is the mutant. The Ames test, once again, is a way of detecting for carcinogenic compounds. So you've got your different strains here. You grow, and you're after exposure, and you're testing to see which ones are potential carcinogenic ones. Genetic diversity. Vertical gene transfer that refers to movement of genes from one generation to another generation. Horizontal gene transfer refers to the movement of gene from one organism to another organism of the same generation. Now, with prokaryotes, genetic diversity, how do you pick up additional DNA? There's 3 main ways. Transformation, you may take up naked DNA that's in the environment. There is DNA out in the environment, so you may pick it up that way. Transduction, this is where you're going to, instead of picking up naked DNA, DNA is introduced to the bacteria by a virus. And then conjugation is when DNA is transferred from one cell to another through pili. Remember, pili are those appendages like a tube that is present with some bacteria. Now, only your gram-negative bacteria have pili, so this is only conjugation can only occur with your gram-negative bacteria. The two cells must be compatible with each other. This is where we have the concern of antibiotic resistance genes on plasmids being transferred from one cell to another. So transformation, this is just showing once again from naked DNA coming from the environment. Transduction is from a virus, and then conjugation is with the pili. This would be the pili right here. That tube that connects so it goes from one to the other. So, excuse me. When you look at genes, we often talk about operons. And what we are referring to with an operon is a segment of the DNA that codes for usually more than one protein, but they're related. So this whole section right here is the operon. It's going to contain the promoter area. Promoter, remember, that is where your RNA polymerase is going to bind during transcription. Usually contains an operator region, and it's in this orientation. And then it contains, here are the genes for the protein that you're talking about. So if transcription occurs, it's going to read A, B, C, and D to make these 4 different proteins. That's going to be necessary. Now, oftentimes what we call upstream, further up is a regulatory gene that's going to help control this operon, turning it on or off. Don't make those proteins if you don't need them. So the operon is that set of genes that's controlled or regulated as a single unit. Instead of controlling each one of those 4 genes separately, you control it as a unit. The lac operon is the most well-studied operon. You have a repressor that normally binds to that operator region when lactose is not present. That operator region was that region between the promoter and the gene. So that's where the repressor will bind. If the repressor is bound to the operator, RNA polymerase can't bind to the promoter, and even if it could bind to the promoter, because the operator is between the promoter and the genes, it can't, the RNA polymerase could not get by it. And so it prevents transcription from occurring. If you don't have transcription occurring to make the mRNA, you're not going to have translation occurring to make the protein, but that's fine because you don't have lactose present, which means you don't need the genes to break down that sugar. Why waste time making proteins you don't need right now? When lactose is present, lactose will bind to the repressor, and because lactose binds to the repressor changes shape and it cannot bind to the operator. So lactose is preventing the repressor from binding to the operator. Nothing's on the operator region now, so RNA polymerase can bind to the promoter. It can pass right over the operator region because nothing's there, and it can go ahead and you can have transcription occur, then followed by translation, and you're making the proteins that are needed to break down the lactose because lactose is present. Now, once the lactose concentration starts to really decrease because you're breaking it all down because you're making the proteins, well, now it's the repressors, you're breaking down all the lactose. So the lactose that was bound to the repressor gets broken down. It's no longer bound to the repressor. Now the repressor can go back and bind to the operator region Well, if it's bound now to the operator region, then that means RNA polymerase can no longer bind to the promoter and have transcription. It's basically shutting everything off, which is fine because you're running out of lactose anyway. So it acts as a wonderful regulatory type of process for turning on and off this operon. So this is showing here the promoter region, the operator region, the LAC-Z, LAC-Y, and LAC-A. Those are the 3 genes that are necessary to produce the proteins to break down lactose. So this is just showing on the top when lactose is not present. Here's your repressor binding to the operator region, preventing RNA polymerase right here. It cannot get through. It physically cannot get through here because for transcription to occur, it has to get through this operator region and it has to read these 3 genes. It can't get there. But that's okay because you don't have any lactose, so why make those proteins? But when lactose is present, like I said, here is the lactose that is bound to the repressor. So the repressor is not bound to the operator. RNA polymerase can just pass right on through, read, and transcribe these 3 genes. You have transcription and translation. You make the proteins. They break down the lactose. Once all the lactose is broken down, then it will come back and be in this situation. Some proteins are always being made, but not all of them. Things like glucose. The enzymes necessary to break down glucose, because glucose is the #1 source that any cell is going to use to break down. All those enzymes involved for glycolysis that we've studied, those are always present because glucose is your primary source for energy. Those are always going to be turned on. If you're given a choice, and I mean you as far as the cell, whether it's a bacterial cell, a fungal cell, or any cell really, if it's given a choice between glucose and something else, it's always going to use glucose first. So those proteins are always turned on. If you're looking at, let's say, in this example for this chart, they're using E. coli, the bacteria. So it's always going to have the proteins available for breaking down glucose. Lactose is a disaccharide, so it has 2 ring structures on it. It's a little bit harder to break down. It can do it, but it's a little bit harder. So if it's given a choice between glucose and lactose, it's going to do glucose first. Ignore the lactose. The enzymes to break down lactose, because it's not always in an environment with lactose, are not normally turned on, as we just saw with the lac operon. So something that we see, it's kind of a cool study to see, if you take glucose and you put it in a solution that has both glucose and lactose, you inoculate it. There's always, on any growth curve, you have your initial, right here, your initial little lag period. Then you start to see the growth increase. This is when it's using the glucose. Then there's a plateau here. Why is there a plateau here? That's when you know it has run out of glucose. And there's a little bit of a delay as it's like, we just ran out of glucose. Oh, but wait a minute, we have lactose here. And then it starts breaking down the lactose. As it starts to level off here, now it's run out of both sugars. It's run out of the lactose as well. But it's kind of an interesting experiment to do where when you graph it out, you see this definite little shift here, this little plateau right here of when it has run out of glucose, and there's a delay before it starts breaking down the lactose then. So I always say there's a fine line between laziness and efficiency. A cell does not want to expend any energy if it doesn't have to. It doesn't want to waste energy. Why would it make proteins to break down a chemical if that chemical is not even present? It's not going to want to do that. So you can argue and say, you know what, the cell's very efficient. and that it's not going to waste its time and energy to produce something it doesn't need. But you could also say, is it lazy because it doesn't want to do that extra work? I mean, if you think about it, there is kind of a fine line between that laziness and efficiency. So, the next time somebody accuses you of being lazy, just say that you're being very energy efficient. You are not wasting energy if you don't have to. But it is interesting to look at these processes of DNA replication, transcription, translation, and the regulatory control that is going on for all of these processes. It's very closely regulated, so it doesn't do things too soon or too late. It's always in monitoring its environment for translation. What proteins do I need right now? I may need to change in a moment's notice. The environment I'm in right now may be very different in 10 minutes. And so one advantage of the operons is it allows the cell to very quickly change to a changing environment. And it's all about survival.